dates confused as well, right?
I did get the dates confused. I was ready to present in 24 hours.
That's why when I contacted you, I WhatsApp you last night and you did not reply. I had a sneaky feeling. I think you might have gotten the dates. Yeah, a little confused because we are already the 15th this morning, but I know back in Canada it's still the 14th, right?
That's
14th
right.
in the evening. Yeah, so it's a bit tricky when we are at different time zones.
So I hope there will not be interference. By strange coincidence, on my street, just in the house across the street, they're making a movie tonight, like a Hollywood movie. And so
Really?
don't ask me why. And so my street in Montreal is representing Philadelphia. And so there's all this noise and these trucks and everything in the street. It's quite funny.
Oh, okay, that's interesting enough. Well, you can't be watching them what they're doing because you'll be busy doing the webinar for us.
That's right.
But it's okay. It sounds peaceful in the background on your part. Sounds all right.
Good.
Sounds
Glad.
okay. So how are you, Joel? It has been like over, I think over a decade since I last met you and Professor Romas as well. How is
Yeah.
he? And how are you?
I'm yes, quite well, thank you. Busy, of course, we all are. And Professor Romas, I have not seen him since before COVID. I was at a few conferences, but I did not see him there. But I have news of him, and he apparently is doing very well.
Oh, that's good. That's good. Okay, that's good. So he's still back in is it MIT? He's
No,
still there?
Minnesota.
Oh, sorry, he's still at Minnesota
Yeah.
University. He's still there. Oh, okay,
Yes.
that's great. That's great. So because I was thinking of contacting you or him, like webinar, but I got hold of you first. So my two professors, which I met during my PhD, and it's great to see that you're doing so well, and you look the same. You haven't changed at all.
So do you.
Oh, no, wow, I'm a lot older, but you look absolutely the same since we last met at the wedding, right? The wedding of my friend's wedding. I think she has 3 children now.
Oh, that's wonderful. That was such a
Yeah.
memorable event. I so enjoyed that.
Wonderful. Our typical Malaysian wedding. Yeah, yeah. So I am glad that you enjoyed all of it. But maybe, Joel, would you like to try and share your slides to see whether it works well? So we have another 5 more minutes. We'll wait about like 5, 10 more minutes for the participants to come in because usually Malaysians tend to be fashionably late. They tend to be fashionably late. But would you like to try your slides? To
Yes,
share your slides?
I'm looking
Yeah.
now. I have instructions.
Mm.
Okay. Plus tard. Okay, that should be good. I think I should be able to now. Hmm.
You can just click share
Yeah,
and
it's
and
not.
then you can use the alternate tab buttons so that you can move from one window to another.
That's not a.
Yeah, just click the share button.
I might need to leave and come back because it's asking me to do so. I don't have the
Okay,
right
go ahead.
system preferences.
Okay. So you can leave or and come back in. All
If I
right.
need to, yeah, it's not,
Yes.
uh, yeah, it's not allowing me. I'll be back in a minute.
All right. All right.
Morning, doktor. Saya tak dengar.
Tanya pada Erna Erna sorry, tanya pada Erna naik PM. Dengar tak suara?
Okay.
Ya, jadi suami isteri dah settle. dah settle tak sempat jumpa hari tu masa scam. Because saya dah ke utara untuk menyelia.
Saya pergi hari
You
kedua.
hari kedua kan?
Saya kedua, hari pertama tak ada bilik.
Oh ya ke? Saya pun,
Alamak,
saya duduk tempat lain.
tak mampu lah, tak mampu. Hari pertama tu sebab kita last minute booking kan. So, bila tengok harga tak mampu. So, tak apalah.
Harga dia tinggi dekat Agoda ke atas booking tinggi ya?
Saya tengok Tibago semua sebab kalau
Oh,
sebab ada family kan. So, mostly kita mesti nak cari yang okey sikit. Macam anak boleh selesa
hmm,
duduk. So,
betul betul.
yang saya cari-cari
Itulah.
tak jumpa. Hari pertama saya tak boleh.
Tak apalah. Tapi uh, apa tu Terengganu was quite full masa saya sampai tu. Meriahnya KT. Ramai cuti-cuti Malaysia. Apa tu cuti sekolah kan? Cuti-cuti
Cuti
Malaysia
sekolah.
tu.
Saya
Ya,
terlupa at
cuti
that time
sekolah.
cuti sekolah. Saya
Yelah sebab anaknya belum sekolah lagi.
tu saya terlupa tu bila last minute saya sebab selalunya saya bila dah nak tempo date tu saya akan booking sebab harga macam dia dah nak murah dah kadang-kadang lah. Tapi masa
Uh,
saya tengok harga naik. So saya aduh.
Memang harga naik cuti sekolah memang melambung lah. Melambung.
Uh-uh.
Tapi cakap terus terang, kalau walk-in lebih murah. If you go through Trivago at booking,
Oh,
you extra by like RM30, RM20
saya
up to
saya tak tahu lah.
RM50 lah. Gitu. Right, aja. So, okay, you can try to use the alternate tab to move around between windows. Yeah, okay, that's right.
That's good.
Yeah, okay. It looks great. It looks great. Maybe you can forward a couple of slides to see whether it's working.
Yes.
All right. Okay, great. Yep, that's good. Good.
I wonder if I can have a laser pointer. Yes, look at that. Good.
All right. Okay. So we'll just wait for about 5 more minutes and then we'll begin.
Should I stop screen sharing?
Oh, it's okay. Just leave the screen on. I think that is good so that when the participants come in, they can see that we are ready. You're ready. Okay.
Awesome.
All right. I'll just give it 5 more minutes. Dr. Azmi, lagi
Yes.
5 minit nanti kita mula ya. Okay,
Yes.
lagi 5 minit.
Cool, okay.
So just bagi peluang untuk
Yeah.
apa tu orang lain masuk. From CT 2000, can I request for the recording of this webinar?
Yes.
The webinar is being recorded. Yeah, Dr. Azmi, right?
Mm.
Is being recorded. So I think analysts will share this. Maybe you can share the recording with me, Dr. Azmi, later, or you blast it on the analyst website.
Dr. Wanil, actually the recording
Yeah.
is we always upload in the YouTube.
Oh,
Because
YouTube.
of sign analyst.
Okay.
So we currently this year we upload all the recording in the YouTube because we want to make sure that analyst is visible in Malaysia. Yes, we know that analyst is well known at Malaysia, but for the other countries currently it's not well known. Yes, I know, but currently we are going to go more. Prof, thank you very much, Prof, for accept our request or our invitation to do
it's my
become
great
a speaker.
pleasure. It's really my pleasure. It's so nice to see Era again. I'm so proud of you for having completed your studies and having this wonderful position.
Thank you. And remember our friend Professor Basharuddin, he just came in. Joel, yeah.
Hi, Joel. How are you?
Nice to see you.
Yes, me too.
Oh, it's so nice to find some familiar friends.
Great.
But I'm still very sad to be doing it only virtually because the food was so good.
Do you know what I always remember about you? Because you said that you like what you call this a toothpaste.
Yes, that's true. That's true. When I travel, for some reason, I always like to go and buy toothpaste that has different flavors, right? And then it lasts a long time. I can remember that country.
That's very good.
I hope ours tasted good.
Yes.
How are you? Assalamualaikum, Probasha. How are you today, this morning?
Good. We just finished our World Forum for Women in Science. So basically, I was the chair for the session. For the whole event, the World Forum International Conference for Women Without Borders. And also today, actually, we are handling the research entrepreneurship, because we are highlighting the research partnership, research leadership, and as well research entrepreneurship, especially for the ladies, for the women in science. Even we got the UNESCO Mustafa Science Technology Foundation to be part of us.
That's
We appreciate the
wonderful.
ladies.
How many participants did you have?
More than 200 actually.
That's fantastic.
Yeah, so we got it all around the world, including not just in Asia, but also in Africa, Europe, South America, and as well. I think in America as well.
That's
Yeah.
very exciting.
Yes.
I think Prof. Joel also has already, I think, embarked on another sort of research, right? Something a little different now, you were saying. Maybe Prof. Basha would like to hear what you have been doing so far, something new that you told me, Joel.
I'm not sure. I don't remember what that was. You mean
I forgot our email chat was quite long, but you forgot, you told me something new. Yeah, something new.
about starting up a
Uh,
company?
something on the company and research as well. Research as well. It's a slightly different angle of research. I think
Yeah, I mean, I guess what I will present today is really the newest and most
Yes,
different
I could say so.
ah,
I could.
because,
okay, all right.
yeah, I had never thought of COVID before.
I also embarked from what you call this, from doing lots of structural biology, metalloenzyme, working more on the for the lung cancer, and also it's actually the fundamental part is more on the designing the nanomaterials for the delivery. Yeah, back to my roots. Still working with the metal, but in such way metal organic framework. So
Oh,
it's
wow.
it's too expensive working on metalloenzyme and structural biology, so I can't cope with that. We don't have enough money. Yeah,
Yeah, and even we've had so many problems for the past 2 years with product delivery and availability. And so molecular biology has just been so difficult.
right. I agree with that. Yeah. Okay.
All right. I think that it's already 9:35 a.m. Maybe we could start now. Yes, Joel? Okay. All right. Good morning to all of the participants, and thank you for attending the webinar. Okay. Which is our regular webinar, okay, done by analysts, the Society of the Analysts. And this time, we are lucky to have our next expert, yeah, who is Professor Joelle Pelletier from the University of Montreal. So I last met her over a decade ago. So it's great to see an old familiar face whom I know when I was doing my PhD. So let me introduce our speaker for today. So following a Bachelor in Science from Ottawa University in 1989 and a PhD from McGill University in 1995 and a postdoctoral fellowship at Université de Montreal and at the University of Zurich, Joelle Pelletier became a professor of chemistry and adjunct professor of biochemistry and molecular and cellular biology at Université de Montreal in 1999, where she holds the Canada Research Chair in Engineering of Applied Proteins. Her transdisciplinary research has produced new enzymes for applications in health, materials science, and industrial synthesis, and has provided remarkable insights into the fundamentals of protein biology. She is the associate editor at ACS Catalysis and has served for 12 years as co-director of Proteo, the Quebec network for protein function, engineering, and applications. In 2015, she launched the startup company Affinity Instruments, where she serves as Vice President in Research. She was awarded the 2021 Clara Benson Award for the Canadian Society for Chemistry, recognizing her distinguished contribution to chemistry. And Pelletier is passionate about mentoring and promoting the young scientists who now lead the new generation of researchers. So please, we welcome you, Professor Pelletier, to deliver your talk this morning.
Thank you very much for that kind introduction. It's a delight for me to be here. Thank you so much for the invitation. It's a bit of a different type of seminar. Well, for one thing, because it's virtual, but also because of the orientation that this network analyst has developed. So I hope that I will properly address the level of interest and of knowledge that is appropriate for what analyst is seeking. And so I will invite, if there are any questions or if things need clarifications, please don't hesitate to ask questions, I guess, at the end of the seminar. So today I'll tell you about, obviously, relatively recent research that we've been doing because it concerns COVID. So just to give a little bit of background, when COVID hit at about the same time for everybody, well, of course, nobody knew what this was. And there was so much rapid development that was done in order to understand what this virus is like and how we react to it. So what kind of symptoms would people develop? And how could we possibly quickly develop assays to detect if people have the disease currently, or even if they have had it. So I'll start telling you about my journey in this and highlight a few special events that happened along the way. So as we now all know, we've now become very educated in SARS-CoV-2 and the COVID-19 infection. We now know that we can detect the presence of the virus by, according to the presence of its RNA, using swab samples. And it's actually quite impressive, the speed at which these highly accurate assays were developed. So we know that there's an incubation period, and I'm sure you've all had experience with either you or friends and family who have had incubation periods of differing lengths. And then we have a period that may be symptomatic, and I'm sure we all know people who have not even had symptoms, even though they tested positive. And then the antibody production begins. So the first type of antibodies that are produced are the IgMs, so the M type of immunoglobulins. And a few days later, the IgGs come up. So the IgGs have typically a stronger production, and the IgG antibodies tend to last much longer, so they can be detected for the longest period of time. Instead, the IgMs, and I will also be showing IgAs, they tend to decrease quite rapidly. So something we might think from this is that if the antibodies decrease, maybe we are not protected. But in fact, antibody production is only part of the immune response. And I will come back to this later. So what we do want to know as questions is, how long do the antibodies last in our bloodstream, in our serum? And also, did you have COVID? So maybe there was a period where you were really tired, but you did a rapid test and it was negative. Well, maybe you did have COVID, but you simply didn't have symptoms, and the rapid test was not sufficiently sensitive to detect the antibodies. So we have to have other types of tests to determine if people have had COVID. So in this presentation, I'll beginning by telling you about a longitudinal study that we did in the Quebec City area. So I have collaborators who are in Quebec City. It's about 300 kilometers from Montreal. And these collaborators work in a hospital setting, a university hospital. So they had access to the clinical staff who could collect samples from volunteers. So we were very lucky to have the participation of 60 adults who had obtained a PCR positive COVID-19 diagnosis a few weeks prior. And so we basically waited a few weeks until they were no longer symptomatic. And then they came for their first appointment. And over 6 months, they repeatedly came to give us their blood samples so that we could follow the antibody progression. The other thing that we looked at is a mail-in study. So the Canadian COVID-19 Antibody and Health Survey sent out envelopes by the mail to 20,000 Canadians, asking them to pick their finger and apply to a paper to give their blood spots. And then you can send this paper back in the mail. So this way, it's a very easy way to collect samples. And I'll come back to this in a little while. So first, we now are all very familiar with this image of the SARS-CoV-2 virus with its little spikes. And by the way, in her introduction, Roswanera mentioned that I really enjoy mentoring. And I'm very, very proud to say that one of my former PhD students, who is now completing her postdoc studies and actually will become a professor very soon, she is one of the people who solved the structure of spike protein at Duke University. So that was quite extraordinary. So the trimeric spike protein interacts with our receptors, our ACE2 receptor, in order to enter in our cells and infect us. And when it enters into the cells, then the viral RNA that is complexed with its nucleocapsid protein can be released. And the RNA will be translated into proteins so that the virus can reassemble more copies of itself. And then as it is producing its proteins to reassemble new copies, our immune system will encounter the spike protein. That actually is immediately, even when the virus is intact, and the nucleocapsid protein following the release of the viral content in our cells. So spike protein and nucleocapsid protein are two potential targets where our antibodies might be active. So if we look again at the progression of the viral infection, we have the virus that enters our cells through our ACE2 receptor. And inside the host cells, this will trigger a whole series, a whole cascade of immune reactions. And so this is a whole long, complicated process that takes a few weeks in order to be completed. And as we progress, one of the parts of this immune reaction is to secrete antibodies. Something else that's very important, though, is these memory cells. And the memory cells are what will help us later on, even when our antibody levels will have decreased, we will still have the memory of the virus if we have been infected or if we have been vaccinated, and this will be able to protect us again. So eventually these antibodies should be able to bind the virus and neutralize it. Neutralizing means that it will remove the virus out of the circulation and therefore allow us to be healthy again. So this is an artist's representation of the virus being escorted out by some antibodies. So the gold standard in the detection of antibodies has for a long time been the enzyme-linked immunosorbent assay, which is also named ELISA for short. So I'm not sure who is familiar with this method, so I will describe it. So what we need to do is, from our target, we need to obtain some proteins. And so in this case, it could be the spike protein, or it could be the nucleocapsid protein that must be produced so that we can apply it into the wells that we will be using for our testing. So here we'll be coating the wells of 96 well plastic plates, and we coat with the protein overnight. And then we will add some non-specific protein. We could use bovine serum albumin. We happen to use milk because of its high protein content. And this is to block some sites on the plastic wells that have not been coated properly with the viral protein. And then we will add antibody. So this antibody, when we began to prepare these assays, we were purchasing antibody to the COVID protein so that we could calibrate our assay. But eventually, this step will be the serum sample from the people that we are testing. So we allow the antibodies to come into contact with the viral antigen, and then we add a secondary antibody, which is linked with an enzyme that allows the addition of a colorogenic substrate and then development of color, which we stop and we measure in a spectrophotometric plate reader. So there are many variations of the ELISA assay. In this case that I'm showing you a colorogenic reaction and reading out in a spectrophotometer for 96 well plates, but there are also versions that have a fluorometric response, and then a fluorimeter is used to read the output. But it's essentially the same construction, where we have a layer with the viral protein, which is the antigen, our antibody that we are querying, and then a secondary antibody that gives the development of a visible response. So here is a typical 96 well plate following the development of the color. So some of the strengths of the ELISA assay are that it's a very well-established assay. So when we came into this pandemic, we realized that this was really the standard assay that we could readily develop specifically for this new virus. It can be scaled up very easily, and it can be automated. So in my laboratory, we already have a robot and some automatic pipetting, some automatic washing that we could apply to ELISA assays. There are some weaknesses though. It's not an instant assay. So it's not like a test strip assay where you get a response in 15 minutes. Here it takes a minimum of 20 hours. So essentially the answer comes the next day. Furthermore, it's quite frustrating for an analytical chemist because it's not quantitative. The response does not scale in a linear fashion with the concentration of antibody. The response is actually a polynomial to the fourth order. So if anybody knows what that means, you must be laughing, because that means that it's very difficult to analyze, and it's certainly not a linear response. Nonetheless, it is semi-quantitative. So we can still tell, for example, with a light yellow color, this is a lower concentration of antibodies, and this in orange is a stronger antibody response. So our immediate objective at this point, we were in the month of March 2020, and COVID was just reaching Canada. And we needed to develop a robust ELISA assay to identify samples that carried the antibodies to it. So as I mentioned earlier, we will be using serum samples and also the dried blood spots. So the advantage of using serum, or it could also be plasma, is that we can obtain from our volunteers very easily plenty of material. So when we take a blood sample, all we need for the assay is a few microliters. So obviously we can get more serum or plasma than that. The disadvantage is that it must be frozen at minus 80 degrees Celsius for long-term storage and also for shipping. So if you are collecting samples from different regions and you need to send it to a centralized test site, this is a very important problem. And that's why we also looked at the potential for dried blood spots to be used. So again, the dried blood spot, you will pick a finger and apply the drop of blood to a paper, and then the paper can simply be dried and put in the mail and sent to wherever the test site is. Very easy. There's no storage required. You just ship it by regular mail. It's not time sensitive. But the question was whether we could use this dried blood sample to recover sufficient material with high enough quality that we could actually analyze the antibodies in it. Now, the secondary objective is, in fact, that we wanted this study to serve as a standard for the development of a very new technology. This is a technology based on surface plasmon resonance detection. It differs very much from ELISA. So what is surface plasmon resonance? This is not as well known as ELISA, so I'll take a bit of time to describe how it works. And it touches on metals, which will please some people in the audience. So what we use in surface plasmon resonance is the properties of gold used at the nanometer scale. So if some of you have worked with gold nanoparticles, you will know that depending on the size of the gold nanoparticle, this of course is not to scale. I'm talking at the nanometer scale. They give different colors in solution. This might seem surprising because we all think of gold as gold, but no, it's actually different colors. And this has been known for a very long time in this form. So in fact, in the Middle Ages, gold was crushed with a mortar and pestle and crushed finely to different colors. And so this, the nanoparticle properties that are, or the optical properties of nanoparticular gold have been exploited for centuries. So how it works in a laboratory is that we have something called a Dove prism. It's a simple little piece of glass that is cut with these angles, and it is coated with a very thin film of gold. So this is really almost a monolayer of gold. And then we have a light source that shines onto the back side of the gold that acts like a mirror and reflects with total internal reflection the light that is then captured by a detector. And depending on what happens on the surface of the gold, it will change the properties of the reflection, and this will change the angle. And when we change an angle, we are changing the wavelength. Therefore, we can see, according to what is happening on the surface of the gold film, we can observe this according to a change in wavelength that we detect. So the change in wavelength will depend on the presence or the absence of molecules that will be absorbed on the gold surface. And it's essentially a mass dependence. That's not quite true, but that's a reasonable way to think of it. So in this case, what we will do is we will bind the antigen of the virus onto the surface. And once it is bound, then we can add a sample that may or may not carry antibody. If the antibody binds, then this will add additional mass, and this can be detected in a change in wavelength. So the advantage of this method is that it's exploratory, of course, but it could be more quantitative than ELISA assays. So this was done with my collaborator, Jean-François Masson at the University of Montreal. And essentially, what it will look like is that we would be able to perform surface plasmon resonance detection of antibodies in crude serum, which is a very useful advance in surface plasmon resonance. Typically, surface plasmon resonance is performed with highly purified samples, but here, what we wanted to do is to use it directly on the serum that is obtained from patients. So here is your prism with the gold surface, and we will add the viral antigen, and then we would flow your serum on, and maybe it contains antibodies that would then bind specifically to the viral antigen. And then if we want to add extra mass in order to get even more sensitive detection, we can add a secondary antibody. So here the secondary antibody is simply serving as additional mass to increase the surface plasmon resonance signal. So I have to say here, I have to make a disclaimer that I own this company. So this is a conflict of interest. Here, we have been using the instrument that we developed about 10 or 12 years ago with my colleague Jean-François Masson. It's a very small instrument. You just hook it onto your laptop for a power source, and you can inject manually your samples, and it will allow you to achieve a reading that will identify the different concentrations as the concentrations increase of a certain analyte. And it's possible to estimate the dissociation constant of the antibody to its ligand, which is an assessment of the quality of the antibodies. Do they bind well? So this system is now commercialized since 2015. So we have been using this instrument. So let me back up a bit, because something that's essential here and that did not exist when we started the study was access to the antigens. So nobody knew yet what the virus was. It was just starting to be discovered. Its RNA was being sequenced, and its proteins were being identified, were being produced. So when we began the project, we were purchasing commercial sources of the SARS-CoV-2 spike protein and nucleocapsid protein, but we had terrible problems with supply, weeks of delays where the precious antigens that should be stored frozen were sitting on a loading dock in transit somewhere in the world with no more ice. And when we were to receive them, we had no idea what condition they were in. And so we realized we needed local sources of the viral proteins to serve as antigens. So with a network of collaborators at the National Research Council of Canada and the National Centre in Electrochemistry and Environmental Technology in Quebec, they developed systems to express these proteins. And we didn't know at first whether these could be readily expressed in E. coli, which is very easy and rapid, but unusual. Typically, viral proteins are expressed in mammalian cells, such as Chinese hamster ovary cells. So we obtained protein from both E. coli expression and from mammalian cell expression, and we compared how they worked, both in ELISA assays and in surface plasmon resonance assays. We were delighted. For one thing, the performance of our locally sourced antigenic proteins was identical to the commercial sources we had worked with. Also, interestingly, whether they were produced in E. coli or in mammalian cells was equivalent, and we could include a polyhistidine tag. This was tested on the nucleocapsid protein, And it made no difference. And this is particularly useful. For one thing, these proteins are typically produced with a polyhistidine tag to allow easy purification. But then often an extra step is used to cleave the histidine tag off so that it does not potentially interfere. But this adds an extra step of protease cleavage that may or may not be successful, and there are always losses of product. So if we can use the protein with the polyhistidine tag, we get better yields of the protein, and it's just faster for our collaborators to produce. It also has an advantage that when we are working in surface plasmon resonance and are using our gold surface, we can actually use the polyhistidine tag to graft onto the gold surface. So these were advantages. Now the spike protein has its receptor binding domain, and this was produced again at different centers, including McGill University. And we got, again, beautiful production from them. And again, different expression hosts were tried. So we had human cells, and an attempt was made to use yeast. So here the thing is that the spike protein is heavily glycosylated, and glycosylation cannot be reproduced in bacteria. That was out. But there are certain strains of yeast, like this Superman 5, that has been developed to imitate the glycosylation that happens in human cells. Because of course, when the virus infects us, it is our own human cells that are providing the glycosylation for the viral proteins. So here, unfortunately, the production in yeast was not successful. It did not give good performance. And this is too bad, because it's much faster and cheaper and better yielding to purify out of yeast. But the protein simply didn't work, probably because its glycosylation was not appropriate. And finally, the spike protein was produced again at the National Research Council, fantastic collaborators, beautiful quality of protein, and this was successfully applied, again, with and without the polyhistidine tag, performed well. And even at every step, as soon as they obtained high quality, high purity protein, a new variant was coming in. And of course, we know now that not only do we have the alpha, beta, gamma, delta, of course, we're at Omicron, and we're at Omicron BA1, BA2, BA3, BA4, BA5, and it seems that there will be many more. So the production of these antigenic proteins is not static. Every time there is a mutation of the spike protein, the process has to be redeveloped, recalibrated, re-verified. And so the proteins that are produced by the National Research Council of Canada are qualified as standards, because they are produced in very large batches that are characterized very well. And so this is a long process, and it has to be started every time that there is a new variant that we want to study. So now we have our system. We can perform ELISA assays. We can perform surface plasmon resonance assays. And we have our antigenic proteins, the nucleocapsid here on the left, the spike here on the right. And we looked at how much we need to dilute the serum, and how what type of reaction we obtain at first with commercial antibodies. So the reason for this was that it takes time to recruit people to give their blood. So when we were establishing the assay, by now we are in March April, May of 2020, in parallel, the recruitment of the donators was underway. And of course, we had to wait for people to get COVID-19 and also to volunteer, to accept to volunteer for a 6-month period. So here what we were able to determine is using commercial antibodies to the SARS-CoV-2, the lower limit of detection for either antibodies to the nucleocapsid or antibodies to the spike protein. So we determined this number, but what does this represent in the serum of humans? Is this sufficient? So I'll show you now some results where we compared the detection that we obtained from dried blood spots, that are reconstituted in buffer, so phosphate buffered saline, and plasma. And what you can see here is that for the spike antigen, whether we are looking at dried blood spots or plasma, both give ELISA signals that where everybody who should be positive is detected as being significantly positive, so significantly different from the negative. So this was great news. And using the nucleocapsid also, all of the positives could be distinguished from all of the negatives, whether we are using the dried blood spots or whether we are using plasma. So this was an excellent advance because it showed that the ease of collecting and shipping the dried blood spots was actually useful so that we could reach populations that are not in major urban centers. They could simply mail in their blood samples and a reliable analysis can be performed using ELISA. So then we looked at the serum of these people. This was, again, all the samples were collected quite early in the pandemic. This was before anybody was vaccinated. And what we are looking at here is the difference in ELISA signal in all these individuals. Each line represents the signal from one person at about 10 or 11 weeks after they obtained their PCR positive COVID diagnosis. So what you can observe here is big differences. So different people might have very strong immunoglobulin responses, or in a few cases, essentially none. So these people, I'm pointing at 3 very low values here. These are people who probably did not produce antibodies, meaning that they did not seroconvert. These may be people who are immunosuppressed. They might have been taking certain medications that made them temporarily immunosuppressed. And so these are individuals who were followed with great interest after, and in particular, when they were vaccinated, to see if after a few exposures, first being sick with the, being infected with the virus, and then being vaccinated with the same protein, perhaps they would then seroconvert. And indeed, that's what we saw in some cases. So even though the ELISA assay is only semi-quantitative, instead of giving a linear response, it does reveal significant differences among the production of antibodies in different individuals. And this is really a key tool now. And again, ELISA is quite easy to set up. So here, what we did, we looked, just to demonstrate that it's not a linear response, this is a 2-fold dilution series. Now, if you have a 2-fold dilution series, you should expect to have a 2-fold change in the signal at each step. And this is clearly not the case. So as I mentioned before, we calculated, and this was verified by other groups, that it's a fourth order polynomial relation. Now, we then had to decide for our individuals what was the appropriate serum dilution that we should apply for everybody, so that we could, at the same time, capture a good enough signal for most people, but also to be able to see who is not developing enough antibodies. And we and this depends on the signal, which is based on absorbance. And as you may know, with the instruments that you use, absorbance is also not linear when you hit saturation of your instrument. And so we had to remain in a region where the instrument could be sufficiently robust but still give us some good differentiation between the individuals. So we chose a particular dilution, which was 1 in 800 dilution of the sera. And we performed repeats, independent repeats. So different plates, 3 different users, different days. And these repeats of the same sample were performed in order to assess the variation of the assay. ELISA assays are quite frustrating. They are harder to control. For some reason, that if somebody has the answer, I would love to know it, but it's been observed by all ELISA users that there is quite a spread in the values that one obtains day to day, user to user. And so the way to deal with this is actually to include a calibrant. So every time you do an assay, you include one sample, always the same one, on each of your 96 well plates. And that particular sample must be within a certain value. And if that sample has a value too low or too high, then you throw away the entire plate. That is the solution. And so we found with time that approximately 5 to 10% of our plates lie outside of the region that we need to be looking in and must be discarded. So initially we were at 30%, and now, you know, this is no longer 2020. Now we are at about 5 to 10% rejection rate. So this really demonstrated the importance of using an internal standard. It's absolutely essential to compare day-to-day variations, even reagent batches. You open a new bottle of one of the reagents. How will it be? The reagent is fresh or it is old? Our shipments of the antigens are different. All these parameters. So now we have these samples from 60 individuals, and these people came at weeks 3 after their diagnosis, week 4, week 8, 12, 16, and 24. So 24 weeks is our 6-month line. And what you're seeing here, the names, HEMA Quebec is the Quebec Blood Services. So they partnered with us. They assessed the identical samples that we also assessed. So here is our assay, and here is theirs. And we actually, at the University of Montreal, we did 2 assays. This is the dilution of the serum that we used. So here it is more dilute, 1/800. Here it is less dilute, more concentrated. So what you can see is the same trend. You can see that at first the antibody response is quite high. You can also see the variation between individuals, where each spot represents the response in ELISA that we obtained for each individual. And after 6 months has diminished, but for most people, we still see very visibly some IgG antibodies. This trend was the same when we used a more concentrated serum. We again see the decrease, but here we get a better identification of the antibodies even after 6 months. And if we dilute more, then we see a similar trend where at week 8 and week 24, week 8 and week 24, so we essentially get the same results that were obtained in our partner institution. So this was important because our partner institution had set up their ELISA assays independently from us. Now, ELISAs are ELISAs. Everybody does the same steps, but we order different reagents from different companies. The reagents that are used for ELISA, there are variations. You might want the one that has this type of acid stop or that type, and different concentrations. Our instruments are different, and the specific ways that we perform certain steps might be a little shorter. We might shake more, all these little differences. And that was very important to us because at the beginning of a pandemic, everywhere in the world, rapidly, everybody has to be developing this type of assay. Can we actually compare them? Or are they too different when they are set up at different institutions? So what we demonstrate here is that we get essentially the same response using the same samples, but with slightly different methods, different reagents at 2 independent sites. So this really validated the robustness of using decentralized ELISA testing to identify individuals who have seroconverted or not. So this is something that will carry on to, the next unfortunate world event we will have. So if we now look at this data, again, we are at the different weeks, 3, 4, etc., until week 24 or 6 months. We see that with age, there are some significant differences. So if you look at the purple, which is the oldest people tended to have a much stronger antibody response for the IgGs against spike than did the younger people who are in this dark gray. And well, we know that the younger people tended to exhibit fewer symptoms, and apparently they have a lower antibody response. And we can see, though, that in all the populations and all the ages, the production is high at first and decreases with time. However, if we compare females and males, we did not see a significant difference. You can see the medians here that are in the middle. We did have fewer men in these in our cohort than women, but there was no statistical difference. If it was statistically different, we would have identified it above with these asterisks. Now, also, an important question is, if you have been infected with one variant of SARS-CoV-2, will your antibodies recognize the new variants that come up? So all the people that we tested, they had all been infected in the very first or second waves of SARS-CoV-2, and that was before any of the variants of concern came to Canada. So they were all infected with an early strain. But then, so I'll call this the original variant. But then we tested by immobilizing the antigen on our surface with the beta, the delta, or the gamma. And what we see is a slight decrease in the response. This is for the samples that were taken at week 4 or again at week 8. We see a slight decrease, but still a good recognition. I will not show the results because they're not fully analyzed, but we have also more recently been testing with Omicron. We are using Omicron BA1, and there the response is about 50% of this. So there is still a response to Omicron by antibodies resulting from infection with the original variant. but it is not as strong. So we can demonstrate by these methods that the antibodies that were elicited in people with the original variant, so the original spike, can recognize the spikes that are produced with variants of concern. Now, I've shown you the humoral immune response against SARS-CoV-2, and I mentioned also that when this happens, that we get antibodies that are formed, and of course the other way to get to the antibody formation is to vaccinate. And we are very lucky to have had, in many parts of the world, very good access to these vaccines. This is a political discussion that I will not get into, but quite good access. So at this point of the study, we obtained funding for a new study. And it happened just as the vaccines were coming in. And so we proposed to look at the antibody development following either infection or vaccination in a much larger cohort. This is 300 volunteers from the Quebec City area. Now, these were people, regular workers, so people who had to continue working throughout the pandemic because, for example, they were working in grocery stores or hardware stores. And we purposely did not look at healthcare workers because healthcare workers have been very studied for very good reason, but it left a gap. What about everybody else who had to work and be exposed all the time to people? Maybe they're getting sick, maybe they're asymptomatic. So that was our focus group. And this study also includes assessment of the neutralization potential of the antibodies. So I mentioned that it's not just about how many antibodies you have, but what do they do? How good are they to escort the virus out by neutralizing it? So again, the challenges were to have access to the virus-specific materials. We needed quality and quantity, and in particular, with the new variants of concern that were cropping up. And also we needed to continue to have access to a constant stream of reagents. And we had several periods of a few weeks where we had to stop working because we were desperately waiting for pipette tips or for the color revelation reagent. So this happened a few times. And of course, always our challenge with calibrating the assay. So here is the timeline of our new study with 300 participants. So I started 2020 just to show the timeline of the which variants were in Canada. And so around 2021, then the alpha, beta, gamma, delta showed up and became dominant. And these lines are when we collect our samples. So the sample collection is shown here. Oh, I'm sorry, this is in French. The word down here means the waves. So these are the waves of COVID that are shown here. We're currently entering our, I don't know, our 8th wave, I guess. So the samples were collected through these, essentially from the 3rd to the 7th wave. And we are still collecting them because initially the sample was to be collected over 6 months, but we were recently extended to be allowed to ask the people to come for a full year so we could get a really long picture of their development of antibodies. So we have a partner here also. We are working with the national facility that is the high throughput serology facility. It's a centralized facility that takes a fairly long time for us to get the results, but the results are standardized. And what we get is a yes or no response, whether people have something called natural immunity, which means did they get infected, or did they get vaccinated? And of course, the difference is vaccination is only with spike protein, but if you're infected, you will also have been infected with nucleocapsid protein, so your antibodies will be different. So here is with the native Wuhan strain at the first visit, and then 3 months later and 6 months later, you can see that we're still analyzing the data here. We don't have all this. And you can see with against the Delta and against the Omicron BA1 that we have good response rates in all cases. And the other thing that we're looking at, as I mentioned, is neutralization. So if you look at the interaction, this is our ACE2 receptor in white with the black glycosylation. And this is the receptor binding domain of the spike protein in red with the pink glycosylation. And whether we can block this interaction with our antibodies. So here we developed a new assay using surface plasmon resonance. And the assay includes the use of the ACE2 receptor that is immobilized. And we verify whether the antibodies of the person can prevent the antigen from binding to the ACE2 receptor that is on the surface plasmon resonance surface. So this is the days after vaccination. So when you have been recently vaccinated, you can very well neutralize the incoming virus. And as the days go on post-vaccination, then typically people are less and less effective at doing so. And this is the same whether we're looking at the Wuhan or the Delta or the Omicron strains. And this was supported also by our ELISA results. Oh, sorry. So that's what I wanted to show you. This is where our second study is now going. So we are, as you can see, we are developing new assays. We are using the literature, all known results as they are coming out to support our studies. And at the same time, what we are doing is to develop methods that can be used in decentralized sites using new technology. And most important is the team. So our COVID ELISA team in my lab, which was rapidly assembled from a bunch of people, Adi Jaileb and Megan Faye Parker have been particularly important to this effort. And I'd like to thank you for your attention, and I'll be happy to answer any questions you may have.
Thank you very much, Professor Kadatia, for the interesting talk. And it seems that those who are doing from of the analytical chemistry background and biochemistry will find it interesting in terms of detection methods and so on. Yeah. So we have already garnered about 4 questions here for you. So the first question, so from Miss Siti, what COVID-2 proteins that you express in E. coli and in CHO? I noticed that both are not of the same weight. So that is the first question. We'll go through the remaining later.
Mm-hmm. Let me see if I can scroll back to that. Here we go. So actually the molecular weight, if you look at the molecular weight markers, they actually do line up quite well. So they have it they just, you know, they're not lined up in the photo, but they actually line up quite well with their molecular weight markers. So they're both around 55 kilodaltons. Does that answer the question?
Right,
Okay.
Miss Siti, does that answer your question?
Good.
Yeah, I hope it does. Okay, right. So that is the first one. So the next question is from Fariza Hanim of UTM. Thank you for your interesting talk. May I know what is the detected concentration of sample when you used the SPR-based prototype? Are they detecting the spike protein? So these are the two questions from her.
Right. So we are not detecting the spike protein, we're detecting the antibodies. We're immobilizing the spike protein to trap the antibodies that may or may not be in the sample. The concentration was functionally the same. Let me see if I have, there we go. So here I'm showing ELISA results, and you can see that this is the lower limit of detection for the antibodies using ELISA. Using surface plasmon resonance, we could go at least an order of magnitude more sensitive, so lower than this. Now, the big question is, what's the concentration in the blood of people? And that's complicated because you have the different type of antibodies. So depending on what we are then revealing by secondary detection, there might be some competition between your different antibodies that are binding, like your IgG, IgM, IgA, that are binding to the immobilized spike or nucleocapsid protein. Does that answer your question?
Well, okay, Miss Fariza, I hope that is the answer. Yes, yeah? All right.
But
Uh,
may I ask more? Like,
yes.
can we compare whatever that we found in this, for example, SPR based sensors to the PCR? Which mean even PCR we can go up until like maybe femtogram per ml in blood. But
Yeah.
then how about we
No.
can compare? Yeah, how to
So
differentiate? Yeah,
PCR is much more sensitive because it's based on amplification of material, right?
yeah, yeah.
Whereas when you're looking for detection of antibodies, you cannot amplify them.
Yes.
So in fact, you're right that the PCR detection is much more sensitive, but it's detecting something different. The PCR is detecting whether you have active virus. Whereas in this case, when we're detecting the antibodies, we're looking either at past infection or at vaccination status.
All right. All right. Okay. Clear. Okay. Thank you very much.
Okay, and there is another question here from Fariza as well. As different variant of SARS-CoV-2 mutate the spike protein, how to ensure the antibody used to detect the SARS-CoV-2 protein will still bind to the sample?
Well, this is an unknown. And so basically, if you were infected in the first wave of the pandemic, the question is now, if you are infected again, will your antibodies be able to recognize Omicron BA6, whatever it is? It seems that there is good recognition. Is it enough to trigger your memory cells and get the whole immune process going again? It appears so. And so this whole question also has to do with vaccination. So vaccination currently uses the original Wuhan strain of spike protein. Is that good enough to immunize us against Omicron, for example? Omicron, which is BA, whatever. Apparently so. And it's actually, it's a bit more complicated than that. Because, so right now in Canada, just since last week, there's a new vaccine that is a combination of the original spike and the Omicron spike that is available. But it's not sure that it's actually better in giving a stronger or longer immunity. So the reason is that if you have already been infected with an early strain, or if you have been vaccinated with the original Wuhan spike. Your body has now developed a recognition for that particular antigen. If you are then infected with a different strain, for example, with Omicron, what will happen is that your immune system will say, oh, this reminds me of the original spike that I was vaccinated against. Good, let's go. And will produce the immune response that is related to the original spike protein that you were exposed to. So even if you get infected by another strain, you still have that, it's called a progenitor response, that the first event that you were exposed to is what will determine your immune response. Apparently this happens from early childhood. So children, the very first immune experiences that they have will shape their immune profile. And so it's very interesting now whether to vaccinate people with the Omicron spike will actually make a big difference or not. I hope that was clear.
Okay,
Yes,
right. Okay, so
excellent.
for researchers,
Yeah, because virus, they are so clever. They know how to tackle in every way. Thank you very much.
My pleasure.
okay. So we have another question from Dr. Norazmi. May I know in this study what are the significant factors that influence the developed ELISA performance? What are the main factors that influence the developed ELISA performance?
I'm not quite, I don't quite understand what you are looking for. Could you perhaps rephrase what you are interested in?
Yeah, Dr. Azmi. Dr. Azmi, can you maybe ask this question yourself? Dr. Azmi?
Good evening, Doctor. Prof.
Hello.
Actually, I want to know a little bit during the preparation for the ELISA. So there are some parameter that we want to develop, optimize. So actually, for the some is some method need some different optimization. So in this case, what is the significant? So that maybe I'm interested to doing this research so that we can collaborate in the next future.
Wow, there was so much that we needed to think of and optimize. So I've got to tell you that before the pandemic, in my lab, we had never done an actual ELISA assay. We had done some other type of similar immunoassays like dot blots or Western blots and such. But to actually use to perform an ELISA assay, so for one thing, you have to get the proper materials. So the 96 well plates are specific for ELISA assays because they have a particular coating that proteins, the antigens, tend to adhere to well. But you have to validate that. You have to validate that you can get reproducibility through your whole plate. So this means, like, is your pipetting system constant? You have to have many wash steps. What is the speed and the strength of the washing, and what is the buffer strength that you will be washing with? And then what is the appropriate dilution of your query material? So in this case, for example, we used serum, plasma, and reconstituted dried blood spots. How much should you use so that when you do your final color development, your signal is not too high that your OD is too high for your instrument and it doesn't mean anything, or too low that you can't make the difference between the positives and the negatives. So we spent quite a while to develop, to have the good color development with the timing that would be very reproducible and that would give us a difference in optical density between, say, 0.1 or 0.2 minimum well, that's for the positive ones. The negatives had to be very, very low. But the highest positives, the people who have a lot of antibodies, we tried to have the response not higher than OD of 2. Although some people still went much higher. And that is, you're beyond the linearity of your instrument. Does that answer your question?
Yes, Prof. Thank you very much, Prof.
Good luck.
Um, may I ask one simple question?
Sure.
What is the difference between, the vaccine, the COVID-2 vaccine, in which after vaccination will last you like it's like a couple of months, about a year, because in Malaysia we've been told to go for booster every year. It was suggested to us. But how is it different compared to the vaccine given to us for like tuberculosis, smallpox? So that is like kind of lifetime. But the one for COVID-2 is short-lived. So actually, being a chemist, I don't really understand this. Maybe you could explain.
Okay, I'm also a chemist. Give me a break. I'm not an immunologist. But here's what I've understood. So, there are really those two very important basic parts of the immune response. What we are looking at here is the antibody production that happens very fast if you're infected or if you're vaccinated. And we look at this, and this is your immediate protection. But the other thing that's much harder to look at is the memory. So if you were infected, as you say, 10 years ago, or vaccinated 20 years ago, is the memory still in your immune system that if you are exposed to it again, it can rapidly increase your antibody production and protect you? I've got to say, I can't give you a proper answer of why with COVID, we are not relying on that long-term memory response. We are really pushing for the boosters and to always have a very high antibody concentration in our blood to protect us. I can't tell you why. If anybody knows, please fill in.
Yeah, that's why it's just kind of interesting, because we are for the COVID vaccine we're going for short-lived short-lived memory cells, and then but for the vaccines, for the smallpox and the ones even for tetanus, it's like I think you're vaccinated one every 10 years. I think it will last for like 10 years. So it's different. But it's okay. But maybe if anyone can answer, we welcome the answer for those who might know. Any other questions? We've already exceeded our time, so it has been really good. Yeah, we have good responses from the participants. Any question, last question that anyone would like to ask? Professor Joel here? Yes?
May I ask one more? Hello?
Yes, you may. Yes, you may.
Farizah here. Hi, Prof. It's just my curiosity, Okay, in normal RTK commercial one, we put our saliva and then they detect whether we are positive or negative. This is my curiosity. What's the level of they detecting the whole virus, right? Inside our saliva. What's the level of detection that usually these RTK have in terms of concentration of samples they require for it to give a positive one?
So here they're actually detecting the genetic material, right? And it's so you have saliva tests that you're doing. It's not like to go up in the nose.
No, just a normal, because commercially nowadays people buy the commercially available RTK, and then we just put saliva on that, and then they tell us whether positive or negative. But then it depends on the day, because if we are already on the 4th day, maybe they are positive, but then at the beginning they are negative. So what's the range of concentration that they require usually in those commercial available RTK?
I can't give you a number. But I can
Oh,
tell you you're really lucky because we don't have saliva tests. We still have to go in the nose. It's really unpleasant.
oh, alright.
Yeah, it hurts. So, you know, there are some people who simply, I don't know why, but they never give a positive response on the test, even though they're clearly like, you know, their husband has it also and tests positive, and they have the same
Uh,
symptoms, and they still test negative. I don't know why.
uh-huh.
Um, and also, like young people tend to give lower response on these rapid tests. I think because they have a lower viral load, typically, right? The children have
Okay.
them.
Uh-uh.
But the actual concentration, I don't know. You might even know, uh, you know, we all hear stories, people who keep testing positive for weeks after they had it. Why?
Uh-huh.
I don't know. So it's clearly it's very sensitive, um, because it's amplified genetic material, right? But I don't
Mm.
know numbers of concentration.
Okay. Interesting. Okay, thank you very much.
My pleasure.
All right. Thank you very much for the last question from Fariza. So we have reached the end of our session. Thank you very much for the wonderful, informative talk and interesting talk from Professor Joel Pelletier. Prof, can we unshare your slide? Because we would like to take a photo session with all of the participants. So all of the participants, please turn on your camera so we can have a photo session for this webinar. Yes. So who is taking the photo? Azmi, siapa yang ambil gambar? Who is taking the photo? So please turn on your camera. Yes. And participants, please fill out the webinar attendance form. Yeah? Attendance form. Yeah, while you're doing so. So are we all ready? Please turn on your camera. Thank you very much. So who is taking the photo?
So I will take the photo, doctor.
Okay, thank you. So tell us when to smile. We are ready.
Okay, all right. Okay, 1, 2, 3. All right, wait a second.
Okay, next page.
Okay, one more. So please, I'd like to kindly request all the participants to turn on your camera, please. Okay, one more time. 1, 2, 3. All right.
Thank you very much, Dr. Hasma. Dr. Hasma was from UTM alumni.
Yes.
Okay, thank you, Professor Pauletta. Thank you very much for the wonderful talk, and thank you to the participants for attending the webinar. All right, with
Thank
this,
you.
Professor Basha, do you want to say anything?
Yeah,
What to say from you?
on behalf of the analysts or the Malaysia Analytical Sciences Society, unfortunately, I'm the president. Okay, so we'd like to thank you again, and we hope that maybe one day we can see you again, either here or maybe in Montreal. Yeah, so thank
That
you,
would be lovely.
Joel.
Thank you so much for the invitation. Thank you for the excellent questions. It was really nice to be presenting with you, and I hope to be able to meet many of you someday.
Yeah, good night. I'm sure that over there is good night already.
Now it's already nearly 11 p.m. in Canada, so I think we will not keep you away from your bed. So, and thank you very much, Professor Joel, and thank you very much to the participants. Okay, bye-bye.
Bye-bye.
Thank you very much, Prof.
Bye.
For your
Thank you
sharing
very
the
much.
session. Thank you.
Thank you.
Thank you also, organizer. It was good. Thank you.
Thank you, Prof.